Brilliant. Okay, so welcome to session 4 on digestion and the liver. And we'd started a bit of these slides last lesson because we were a little bit ahead of schedule. So what we were looking at are the immune factors and the removal of pathogens from the blood. So I went over the sort of outline of the immune processes of the liver. And what I thought I'd do is go into a bit more depth because we were a little bit ahead of schedule. We've got time, so we can go into some more detail about the immune factors in the removal of pathogens in the liver, because there's quite a lot going on there. There's lots of specialized cells, so it's a really important organ. So we talked about the fact that the blood that was coming from the hepatic artery was flowing over these specialized Kupffer cells. They're phagocytotic. They're the grey ones on the diagram. You can see here, they've got the pseudopods. Remember the sort of fake legs, fake arms that, let me put it into slideshow view. That would make it a bit easier to see, wouldn't it? There we go. We've got these projections really, which can be used for phagocytosis, which we've talked about before, which is when a blood cell engulfs a pathogen and then breaks it down. So that's basically what we looked at, and we're on this section here, outlining the production of immune factors, the removal of pathogens in the blood. So we know that the liver is also involved in detoxification, and we had a look at that last session as well. So key points: the blood's coming straight from the gut, which is an exchange surface. So we might have taken in all sorts of toxins and pathogens as a result of that. And we've got these specialized cells, the Kupffer cells, and we've got specialized cells around here as well, the sinusoid cells. So the Kupffer cells, as we said, specialized macrophages, and they're found in along these endothelial membranes of this sinusoid. And those endothelial membrane walls are specialized as well. So the Kupffer cells are engulfing and destroying the phagocytosis that we looked at. These organisms, microorganisms that come from the hepatic portal artery, but also they've got a role in aged or damaged red blood cell removal. So what these Kupffer cells do also is secrete cytokines, which have got quite an important role in activating immune cells, other immune cells. The immune system is incredibly complex. It's incredibly interrelated. I will share a video link for you if you want to have a look at this further. But there are some good videos by a company that work on trying to simplify the immune system, which I can share with you via Google Docs or something. I'll send you the link so you can have a look at that. But it's incredibly complex. And these cytokines are one of the structures, or one of the things basically that interacts between many different parts of the immune system. So they activate immune cells, they promote inflammation, and they also help to initiate tissue repair responses. They can also cause autoimmune diseases to happen if they go a little bit wrong. They can cause a too big of an immune response, too strong of an immune response. So they're really key within linking this system together. So we also talked last session about tolerance to gut antigens being an important part of these specialized cells, so we don't have too big a reaction and end up having something like autoimmune diseases. These are really a key of these specialized cells here. So Kupffer cells filtering the blood, removing pathogens by phagocytosis, producing cytokines to regulate the immune system, so to cause other things to happen, inflammation, for example, and also maintaining immune tolerance to prevent overreacting. So they're defenders, the phagocytosis, and regulators, so those cytokines being produced. So this is getting on now to the bit that I wanted to go into a little bit more detail about. So the liver is filtering this blood, which is potentially contaminated if you've happened to have eaten some bacteria with your food, which is incredibly common, as we will find out in the next unit. No, next but one unit, when we start talking about infections. It's incredibly common to accidentally eat bacteria, especially if you're eating with your hands or if somebody's been preparing your food with their hands that haven't been washed. So the liver, I told you this last session, but didn't go into any detail, is the site of antigen presentation. And this is basically what kicks off this whole immune response. So the liver's a site of antigen presentation. There are other sites as part of the immune system, and we'll learn about that when we cover disease in the next but one unit. But we're going to have a look now, a first look at this system in these specialized cells. So we've got these Kupffer cells. They're attached, they adhere to this endothelial walls surrounding this vein here or vessel here. So often the immune system cells, or usually I suppose the immune system cells, are circulating. So that means they're in the blood, they're floating around in the blood. So the white blood cells are circulating. Other parts of the immune system are circulating, things like platelets and red blood cells. So it's not often that we get immune cells that are attached or fixed, but these are. But they can still engulf and destroy all of these organisms that are coming along. So I'm just going to show you the process. I've just got to move a little block on my screen. It's one of the joys of Zoom, that is. So antigen presentation is what we're looking at. So what happens is antigens are presented to special white blood cells, and then this can kick off an immune response. So there's a couple of things we need to know there. What's an antigen? Well, it's something that you might find on the surface of a pathogen. This is a little picture of a virus. You'd have been used to this picture when COVID was around. These little sticks that are sticking out here are specialized proteins, and there'll be proteins that we don't find in humans. They can get broken down into bits of proteins, and bits of proteins are called peptides. It's just what we call chopped up proteins, peptides. So these viruses are covered in virus proteins. Bacteria are covered in bacteria proteins, and we don't get them in humans. And what this macrophage will do, so it will come along and phagocytose, it engulfs it. We're used to that, we've been seeing that quite a lot. So here it is, it's this macrophage, white blood cell, it's a type of white blood cell, a macrophage, has phagocytosed this virus. You can see it inside now, it's engulfed it. And what it does then is it presents the components, literally shows the components of the pathogen, of the virus, to this cell here. This is a T lymphocyte, just another specialized white blood cell. Nearly all of these cells that we're looking at are specialized white blood cells. There's loads of them, and they've all got varying defense roles, slightly different defense roles. So virus, special virus proteins on the outside, don't get them in humans. The virus proteins, bacteria have bacteria proteins, and we call these antigens. It's just the special name we give to these virus proteins, bacterial proteins, fungal proteins, anything that the body can recognize as non-human. Virus engulfed by this special white blood cell called a macrophage. By phagocytosis, it's now inside the specialized white blood cell. And it is going to show these bits of protein, because if you look really closely, you can see that the virus has been broken down a little bit once it's inside the macrophage. And it can show these protein segments, these bits of proteins called peptides, to this T lymphocyte. So we're going to have a little look at that. This can happen inside, inside the liver as well as inside some other immune organs. So this is really important, a really important process. So this process, we're going to ignore all of this over here, really. We're going to go on and have a look at that later when we talk about disease a bit more. But this process here, where we've got this macrophage, which has got this virus inside, which has been broken down, is what's going on over here on this diagram. It's a little bit clearer because this is, looks like someone's art project, really. This is quite a clear diagram. So let's have a look what's happening. This is our white blood cell, this green thing here. That's our white blood cell. It's the antigen presenting cell. So it's the macrophage, which has engulfed the virus. And here we have the antigens. So bits of the virus have been, basically the virus has been broken apart, and this white blood cell has broken apart all the proteins that made the virus. And it's got this special structure here, which is called the major histocompatibility complex, which is a mouthful, but don't worry about that particularly. It's got a special structure here, which can attach these antigens. Now, there are loads of these, so they can attach all the different type shapes of antigen. Then what it will do is it will show that antigen, which is that bit of virus protein, to this specialized white blood cell here. So let's have a look at this bit here. The red bit is these specialized major histocompatibility complex structures. We have two classes, class 1 and class 2, and they basically are specialized for different things. So one of them will find antigens from things inside the body, so things that go wrong, like tumors and things like that, or infected cells. So a virus has got in an infected cell, and the other group they've got a slightly different role. They find things from outside, like bacteria or foreign particles. Viruses can get inside, well, they've got to get inside cells, really. They don't survive very long outside the cells. They've got a special membrane that gets them inside the cells to copy themselves. So we basically, these red bits here are specialized for two different jobs. Breaking up stuff that's found inside the cells and breaking up stuff that's found outside the cells. And there's loads of them in each class, so that we've got loads of different shapes of these major histocompatibility complex structures, so that we can attach all the different shapes of these antigens. Because if you think about it, there's going to be loads of virus proteins, loads of bacteria proteins, because there's loads of different types of viruses, loads of different types of bacteria. So they've got loads of different shapes of proteins. So we need loads of these so that we can match up and show these proteins to these other specialized white blood cells. So we're taking the cell surface proteins from these viruses, and we're taking the small fragments essentially to the T cells, the specialized white blood cells. And once inside the cell, over here, cells are broken down. This is a breakdown of what happens. The cells are broken down. Yep, so the white blood cell, the green, into these small peptides, antigens, and they're loaded onto these specialized molecules, these histocompatibility complex structures. Then the peptide and major histocompatibility complex is transported to the cell surface, which is what we've got here. The red bit is basically holding this viral protein in place. And then this T cell, which is a specialized white blood cell, will scan this, both bits of this, and decide if it's something to worry about. Is it foreign or is it self? And if it is foreign, it will activate an immune response. So this process is called antigen presentation. It's essentially the process by which the antigen presenting cells, which was that green one here, that's presenting the antigen presenting cells, display the antigen fragments, the peptides that have been broken down from the structures making up the surface of the virus on the histopathology complex molecule here. And they're showing it to the T lymphocyte to see if it's going to initiate a big immune response. So these are the specialized cells, the Kupffer cells that we talked about before, the endothelial cells, and the hepatocytes. The hepatocytes, so they're like functional cells in the liver, the ones that are doing the detoxification and stuff like that, making liver enzymes. So there's lots of specialized cells in here that are important. So the Kupffer cells are specialized macrophages, as we said, they're lining these epithelial cells and really efficient at doing that phagocytosis, engulfing these pathogens. And these can express both types of these major histopathology complex molecules. So both of the, if we go back a little bit, both of those groups there. Oops, where did we go? There. So both classes, so the Kupffer cells, those liver cells, can express all of those types of antigens. So from tumors, from bacteria, from infected cells that need to die, abnormal cells, viruses, anything. They're really good at basically being very versatile. So we've also got these endothelial cells lining the hepatic sinusoids here. And these are able to capture soluble antigens from the blood that are just sort of floating around and present them via their major histopathology complex molecules. Right, just 1 minute, I can see there's a question in the chat there. Oh, I missed one there, sorry about that. In what cases an immune response does not happen? Because if it's still an antigen, would it not be risky? Right, yeah, so that's a really hard question because the immune system is so incredibly complex. When I show you this, I'm not going to show it to you now, but when I give you this video, you'll have a look at it and you'll be like, right, okay. So I think you're sort of saying, when does an immune response not happen? Well, bacteria, for example, can move around in body fluids. So that might be in the blood or it might be in the tissue, but they can move around from place to place. So they might take quite some time before they get noticed by a circulating white blood cell. So if they're going through the liver, where we've got these specialized Kupffer cells adhering to the cells of the other cells of the liver, they'll probably get caught, hopefully get caught, and we don't end up with like really bad infections. But if they've got into the body some other way, for example, through a cut, or they probably usually get caught in the lungs because there's lots of adaptations there as well. But imagine they've got in through a cut and they start circulating in the body tissue, then they might not come across a cell that's able to engulf them for some time, and they might be able to take hold and make an infection start. And so that's one way we can get an infection, even though we've got all of these circulating immune cells. It is just chance whether the right type of immune cell can catch the pathogen once it's got in. And if it's a virus, it takes a while before they get caught because they have to get into the cells really quickly. But when we go through not the next unit, but the unit afterwards, when we start to look at disease, you'll see that when viruses get into a body, they pretty much just get in and get straight into a cell, that you don't see them floating around in the tissue. So there's nothing to detect a lot of the time because they're inside the human cell replicating. It's only when they burst out and get to other cells that they'll have antigens on their surface and things like that. So there are times when we won't notice that we're harboring an infection, unfortunately, or it just manages to get too big too quickly before we can keep it under control. Okay, so soluble antigens then might be in the blood. And these can get detected by these endothelial cells in the liver. And then these help to contribute to immune tolerance to harmful gut-derived antigens. So just to calm things down, so we're not having massive immune responses to stuff that's harmless. So the hepatocytes, so specialized liver cells doing specialized liver things like deamination that we talked about last time when we were taking the groups off the amino acids, the chemical groups, or detoxifying alcohol. So these will express only the one group of these molecules, these major histopathology complex 1. So these are the ones for infected cells, tumors and stuff, rather than this group here, which is more to do with the bacteria. So they can directly present antigens to these specialized T cells, and this will lead to T cell deletion or tolerance in non-inflammatory conditions. So what that's saying is, if there's no need for a big response, it will be kept calm by deleting these T cells if they're not needed. One thing to say is during infections like hepatitis B, hepatitis C, these hepatocytes become targets of cytotoxic T cells, of your own immune cells. So they end up getting destroyed because of your own immune cells and how they respond to these hepatitis B or C viruses. So hepatitis is a liver inflammation. So anything that says itis means inflammation of. So hepatitis is an inflammation of the liver. And these specialized cells that have a major histopathology complex role expressing antigens can become targets of your own immune system during infection. So obviously that's not very good. So here's just a table showing some of these special cells in the liver, the Kupffer cells, the endothelial cells, and the functional hepatocytes. They've all got these varying roles for presenting the antigens to the white blood cells, which will then decide whether to mount a big response or not. And that's just a brief summary of their roles. So the Kupffer cells, they can phagocytose. We talked a lot about that last time. The antigen presenting cells also. The endothelial cells can detect the antigens that are soluble. And then we've got these specialized functional hepatocytes, which present to these special CD8 plus T cells. And note that they all tolerate, or they all promote tolerance of the immune response. So calming things down if there's not a big problem with the proteins that they find. So keeping the response proportional to the danger. However, this can go wrong. And we're going to have a look at an example. This is an example to do with the digestive system as well, since we're on the digestion in the liver, but there are loads. So are hepatocytes epithelial or connective? They're functional, so they're called parenchymal cells. That means they are the cells that do actual jobs. They're specialized cells that do specialized jobs. If they're from the liver, which is an organ, should they not be in situ by default, or not all epithelial cells stay in the same place? Well, they are in the liver. They're the specialized liver cells. So some cells were circulating that I was talking about. They were like immune cells. But I know what I think you're confused about is the antigens can be circulating, not the cells. So the hepatocytes stay where they are, but the antigens might be circulating in the blood or the tissue fluid. Okay. So if this process goes wrong, and for example, the cytokines which are triggered or the response from those specialized T cells that are having the antigens presented to them, if that goes wrong, and we're pretty sure actually that it's due to a cytokine response. There's been some research out today actually suggesting that that is the case. There's some cytokines called interleukins that can trigger this autoimmune process. So there are several different types of autoimmune disease. You might have heard of lupus, you might have heard of Crohn's, colitis, there's loads, psoriasis, loads and loads. We're going to have a look at colitis because it's an autoimmune disease of the gut, and we're on a digestive system and liver section at the minute. So there are several types of colitis, and remember, anything itis means inflammation of, and colitis is inflammation of the colon, which you might remember was another name for the large intestine. So there are several conditions that can cause inflammation of the colon. There's infectious colitis, which means you've got an infection and it's inflamed. And there's ulcerative colitis. It says cause not known. It's an autoimmune disease. And we can't for definite say why autoimmune diseases happen, but we're pretty sure it's when this immune response goes wrong, usually triggered by an infection. Crohn's disease, which is inflammation of the small intestine rather than the large intestine, again, autoimmune, and that is caused by probably an infection triggering it. Ischemic colitis, that means there's not enough blood getting there, so that's not autoimmune. Infectious colitis is not autoimmune either, but these two are ulcerative colitis and Crohn's are both autoimmune. You can get inflammation after radiotherapy, and the symptoms are really not pleasant. It causes diarrhea, bleeding, cramps, frequency and urgency, needing to go to the toilet. Treatment depends on what's wrong, but these you can end up taking immune suppressing drugs to suppress the autoimmune response, which is pretty unpleasant because you're at risk of other problems then, because if you suppress your immune system, you remember some of those major histopathology complex proteins were also looking for tumors and cancer cells. So by taking drugs for autoimmune diseases, you increase your susceptibility to things like cancer. It's not very nice to diagnose. You have to have colonoscopy, which is tube up your digestive tract or down, depending on where it is. But these are at the end of the digestive tract, so they go up through the anus, which is really unpleasant. So it's a pretty horrible condition with pretty nasty diagnosis system. So the immune system, as we were just looking at, is essentially, well, not really designed, but evolved to attack foreign pathogens like bacteria. And as we've just been talking about quite extensively, it's meant to ignore and tolerate the body's self cells. And if it's not a big infection or there's nothing major going on, it's meant to have a proportionate response. But if the self-recognition fails and there has to be some sort of trigger as well, like a big cytokine storm, it's called, when the cytokines get out of control. It can trigger an immune cell, like autoimmune colitis or ulcerative colitis and Crohn's. And what happens is the body will misidentify the self tissue, like your own tissue, or normal gut microbes, because we've got an awful lot of microbes that live in our guts, which are really important for helping us break down our food, and have a most likely massive role in regulating the immune system, but we don't understand that fully or even partially at the moment. So the immune cells that get overactivated in this process are particularly T cells, macrophages, and B cells. I haven't talked to you about B cells yet, more about them in the future units. So these are the T cells, the ones that we're presenting the antigen to. These are the macrophages, the ones that do the phagocytosis, and the B cells are down here. These are the ones that give you immunity. Later on, we'll talk about them as well. So the B cells actually produce antibodies too. So that you might have heard about antibodies, I expect, before. So these immune cells all release these inflammatory cytokines, and inflammatory cytokines cause inflammation. So you get chronic inflammation, ulceration, which is like sores, and tissue damage in the colon, which is really painful. You can see the damage to the tissues. And essentially what you have in colitis is unusual major histopathology complex expression on the epithelial cells or the dendritic cells, and it can lead to this autoreactive T cells being activated instead of giving the tolerance and calming that response down as it should. Okay, right. Any questions before I go on to this next bit? We did a little bit of this bit last session. So I'll go over this first bit that we talked about last lesson quite quickly. So we've talked about how the hepatocytes clear the blood of drugs and other toxins. So we talked about how it removes the group of the amino acids to break down proteins, and it can recycle those last session as well. But as well as clearing up things that we make in our body, so in other words, metabolic waste, it does clear up stuff that we consume. So it detoxifies drugs, alcohol. What drugs could mean, you know, medical drugs, it's not just recreational drugs, because they often have quite a lot of side effects as well and need to be broken down. So the hepatocytes, the specialized liver cells, play an important role in metabolizing these substances, drugs, alcohol, and waste. So obviously all of this stuff is consumed orally, goes to the digestive system. First place the blood goes from the digestive system, hepatic portal vein, straight to the liver. So we spoke about the fact that the smooth endoplasmic reticulum was important for these detoxification reactions, and it's a very energy hungry process. So the liver has a lot of mitochondria as well. There's a lot of enzymes involved in these oxidation, reduction, and conjugation reactions, so adding together and breaking stuff down. So we had a look at this structure in the first session, and this is the smooth endoplasmic reticulum in blue in the middle, slightly different color to the rough endoplasmic reticulum. So the rough endoplasmic reticulum has got these little yellow dots here, ribosomes attached. That structure is here in black, continuous with the nuclear membrane, so joined onto the nuclear membrane. And these are the sites of protein synthesis. So we talked about the fact that the DNA is the instructions to make the protein. This is the site of protein synthesis. But over here, we've also got the smooth endoplasmic reticulum. And in the liver cells, this is important in detoxification. It's also got a role in lipid synthesis and calcium storage as well. So what happens in the detoxification? Well, it's the first phase is modification. So this could be oxidation, reduction, or hydrolysis of toxins and drugs. So essentially, these are all chemicals that are being chemically changed. When you do chemistry, you'll learn that about these processes. So I'm not going to go over them here because they're quite detailed and complex. Just the name is okay at the minute. They get changed, they get modified, and these reactions are catalyzed by enzymes. So enzymes are structures which speed up reactions. So there's special chemicals, proteins essentially, that speed up reactions. And what they are doing is they're changing the chemistry so they can remove groups from the chemicals. So OH groups or NH2 groups, for example. And this changes how the chemicals, the drugs, the alcohol behave then. So for example, alcohol is converted by a special enzyme from ethanol to acetaldehyde by a special enzyme. The next thing that happens after these chemical changes have been made to the drugs, so they've had chemical groups chopped off them, etc., is conjugation. And conjugation means to stick something together. So hepatocytes will then attach another molecule to the modified compound. So the chemical that they changed structurally, they'll stick other stuff to it. What this does is it increases the water solubility, makes it a lot easier to get rid of this compound. What you've got to think of is we're doing everything we can to get this out of the body because it's something that's bad for our cells. So alcohol is toxic to all of our body cells. The drugs, depending on what they are, that depends on how bad they are for us. But paracetamol is an example of a drug which can be quite toxic, and that is one which is conjugated or stuck together with another chemical, glucuronic acid or sulfate, to get rid of it from the body, to break it down, to detoxify it. If you take excess amounts of paracetamol, it can overwhelm this pathway, so the pathway can't cope quick enough, and it can lead to toxicity. That's why you're only ever sold packets of paracetamol in 16 at a time. They won't give you, if you try and order more packets from like Tesco's or something, they will only sell you 2 packs at a time because there's such a high risk. It's so easy to overdose on it. It's a really dangerous drug. So the other thing it does is excrete metabolites. These are things that are produced in the body. So we spoke about it earlier on when we were saying that excretion. I think we were talking about digestion. I think in 2 or 3 sessions ago, we were talking about digestion of waste from the digestive system. We're saying that we don't call that excretion because it's fiber that we've eaten. But here we're looking at excretion. This is a product of our metabolism. So it is a product and a chemical that we've made in our body. An easy example to think of is carbon dioxide that we excrete via the lungs. We make that when we are using energy in our bodies, and we excrete it. It's made in our bodies. So what's going on in the liver is we are creating a substance called urea, and then that is released into the blood, goes all the way around the body until it gets to the kidneys, and it can be excreted in the urine. Some stuff is secreted into and removed in the bile. This will then get excreted into the small intestine. Remember, the bile is produced in the liver also. It's stored in the gallbladder and secreted via the bile duct into the small intestine. So everything that moves through the small intestine is secreted in the feces. So stuff from the liver can either go back into the blood or into the feces for removal. Additional detoxification functions are, which we've talked about last session, the deamination of the amino acids producing ammonia, that's urea, and that's how we get rid of that. Okay, so we have had a look at clearing the blood of drugs and toxins and a more detailed look at the immune factors in the removal of pathogens from the blood. So have we got any questions for me? Just see one there. Let's have a look. Oh, two there. I must have missed one earlier, sorry. I have one from last session. Okay, I'll if you don't mind, I can send you, I'll get back to you on that one, and I can pop those slides back up because I can't remember which slide pack they're in. I can pop them up next session. And I'll move on with the next slides, okay? Uniporters and active transport, I'll remember that. Okay. Right, so next topic, cardiovascular system. So the cardiovascular system is the heart and the vessels that connect to it. So that's things like veins and arteries, and they connect to arterioles and venules, and they connect to capillaries. So it's all the vasculature and the heart and the blood that circulates through it as well. Now, we've met loads of the blood cells already because we've been talking about the immune system, and most of that is circulating around the body. So all of those T cells, we've been talking about macrophages. Don't think we talked about killer cells, did we? No, maybe not. So T cells, lymphocytes, macrophages, all of those are circulating in the blood. So they're part of the cardiovascular system. So we've met quite a lot already. So first job is to understand the cardiovascular system. We're going to have a look at this. It's the cross section of the heart. So what we've got here is the epicardium, the myocardium, the endocardium, and the supporting structures. So you can see the heart wall there. But major tissue types being epithelial, connective, cardiac muscle, endothelial, and we've got nervous and vascular structures as well. So there's lots of different tissue types making up this heart. Now, normally if someone said to you, what is the heart? You'd say, well, it's muscle, it's cardiac muscle, but there's endothelial, connective tissue, nervous tissue, and vascular tissue as well, all with different functions because this is an organ. So we looked at, in Unit 1, we looked at tissues being groups of specialized cells, and we said that they work together to form an organ. And these are all the different tissue types that are working together to form this specific organ. And if you have a look at this image here, which is an old-fashioned drawing, it's there to show you, I think it shows quite clearly all the different structures because it's detailed rather than this drawing here, you can't really identify the different structures. This one here, you can see the different textures. So you can see the vessels supplying the actual, this is the heart muscle itself down here in white. And this bit here is part of the heart as well. These vessels are supplying that muscle with blood. And then these really huge vessels here are the ones that take the blood through the heart for pumping. And then you can see the nerves come in to and from the heart as well. So really complex, loads of different tissues, all working together for a common function. So the function of the heart is, as is well known, is to pump the blood around the body. All these different tissues working together for that function. So we're going to have a look now at these structures. Specifically, we've got the epicardium, which is part of the outer layer. It's made of epithelial tissue and connective tissue. And it's a thin outer layer, and it's sometimes known as the visceral pericardium. What it does is it protects the heart and it reduces friction as the heart beats. It also contains the blood vessels, the lymphatics, and the nerves. So when the heart beats, it moves quite powerfully, and there needs to be a certain amount of lubrication on the outside. There's lots of your lungs as well, particularly. They're another organ which moves around a lot. So all of the outsides of these organs need to be lubricated, or otherwise they're all going to be rubbing against each other and it's going to cause significant amount of pain. So there's lots of layers of insulation and lubrication inside the body cavities. Right, then we've got the myocardium, which we can see here. And this is the cardiac muscle tissue. So it's a specialized type of muscle. So it's not the same as the skeletal muscle. We talked about that a little bit earlier, and we'll talk about that loads more in another unit. But remember we said it was stripy and it had those special proteins that pulled in. This is a little bit stripy, but it doesn't look stripy under the microscope like the skeletal muscle does. It's got a different function, but it's specialized. So this myocardium is the thickest part by a long way. And that's because the muscle is thick and strong. The left side in particular is very thick compared to the right side. It has to be very strong because it's the side that pushes the blood all the way around the body. The right side only pushes it to the lungs, so it's nowhere near as thick because they're right next to the heart. But the left side has got to push it all the way down to the end of your fingers, tips of your toes. So what's the function of the muscle? Pump the blood around the body, and it also contains intercalated discs for synchronized contraction. It has to contract in the exact right order, starting from the exact right place. You can't just have that muscle just randomly contracting. It has to be very carefully controlled so it pumps rather than, you know, the top bit squeezes, then the bottom bit squeezes. That can't happen. It's all got to happen in order and sequence. So it's a very specialized tissue. Then we have got the endocardium, which is the inner layer. You can see it over here, really thin on that side. Specialized simple squamous epithelium and connective tissue. So remember when we talked about the epithelium and we said these were really flat squamous epithelium, really flat cells that looked like tiles almost covering an area, and they were sort of squarish in shape. Another really important thing about those is they've got really tight junctions as well between them, so nothing can really get in between them. And they give a nice smooth finish. What's important, if you have a look at this diagram up here, these are lining these inner layers of these chambers within the heart. It's really important that the blood moves smoothly through there. We don't want it whirling around. We call that whirling turbulent flow. We don't want turbulent flow. We want it to just smoothly go in and out, because we basically need it to flow through so that we have a heartbeat and it pumps. So these lovely smooth layers of specialized square epithelium cells will prevent the blood from mixing around and whirling around. It will just smoothly move through, and it will also help prevent clot formation inside of the heart itself, because we obviously don't want that happening. So the heart itself has got supporting tissues, connective tissues, nervous tissues, and blood vessels. As you can see here, those are the tissues we talked about. So in addition to those layers that we just considered, we've got the connective tissue, which is the sort of skeleton with valves, and it anchors the cardiac muscle fibers. The nervous tissue, you can see there in yellow. This can help regulate the heart rate. The heart is, those are nerves actually, but there is nervous tissue in here as well. It's specialized heart tissue rather than nervous in a way. It's where the heartbeat originates from, and it can well, we're gonna have a look in a bit, but it can change polarity. So it's, in a way, that's not really nervous in a way. It's specialized, but it's similar to nervous tissue. But we have got nervous tissue on the outside, and we've got obviously blood vessels which are supplying this muscle itself with oxygen, because that heart has got to contract because it's a muscle. So why is all of this important? Well, it's because of respiration. We need to get glucose and oxygen around our body so that we can make something called ATP energy. That's how all of our cells do all of their jobs. It's really important. So respiration, we're going to talk about that later in more detail, is releasing ATP energy. Don't worry about that for now, but it's releasing the energy from glucose. Oxygen is usually needed. You can do it without oxygen, but it's better if you do it with oxygen. So why do we need a circulatory system? Well, respiration in our cells needs glucose and oxygen. Glucose is where we're getting the energy. Oxygen is needed to release the energy. And we've got these specialized exchange surfaces. The lungs is where we get our oxygen from, and the small intestine is where we bring in the glucose from. We learned about this exchange surface of the small intestine last week. And the lungs are where we get the oxygen for this reaction from. So the circulatory system, or the transport system, if you prefer, will deliver the glucose and the oxygen to all of the tissues, all of the cells within these tissues. It also takes other things like heat. It distributes our body heat because of the temperature of our blood, and hormones. So it will also take other molecules around. We're way too big to rely on diffusion from these molecules to get from place to place. So we have to pump it around because we're huge multicellular organisms. And we also need to remove all of our waste metabolites. So the carbon dioxide that comes from the breaking down of glucose to release that ATP energy has to be removed, and because it makes our blood acidic. And we have to remove that urea that we were just talking about a moment ago, which the liver breaks down. It has to be pumped from the liver to the kidneys so it can be removed in our urine. So the functions of the circulatory system then are oxygen and carbon dioxide transport. So oxygen in through the lungs, into the blood, out around to the tissues. We get this carbon dioxide produced as we produce ATP, and then that has to get pumped all the way back to the lungs to be removed. So we say nutrient and waste, basically, and CO2 and urea are examples of waste that need to be transported. It's important in temperature regulation because the blood is what basically moves the heat around our body. Hormone circulation, which we'll learn about later on in another unit, and defense, which we had a little look at today, and we'll look at more detail in a later unit as well. So the white blood cells are really defense orientated. So we have three components of the system: the heart, which is a pump, which we just had a brief look at. We've got the blood vessels, which are all the tubes through which the blood flows, and the blood itself, which we've spoken about in an immune way so far, but not in a transport way. So we're going to start having a look at the structure of the heart then. So what you can see on this diagram is the heart. And the first thing I should tell you here is there's a line through the middle. If you can look at this line, you can follow it through. It divides the heart in two. That's called a septum. That's the division between the two sides. So this is the right side, and this is the left side, which you might have noticed if you're paying attention is not the left and right side of the screen. So this is the left side of the screen, obviously, and this is the right side of the screen. But this is the right side of the heart, and this is the left side of the heart. That's because you're looking at the heart of the patient who's on the operating table in front of you. Okay, so that's why it's back to front. So patient's in front of you, so this is their right side, and this is their left side. So the heart has got 4 chambers. This is the top chamber on the right side. This is the bottom chamber on the right side. This is the top chamber on the left side and the bottom chamber on the left side. And it has, well, it has veins going in and arteries going out. And on this diagram, we've got the veins labeled up. So we've got the vena cava, or some people call it the vena cava. And both of these are labeled vena cava. And that's because you've got one coming from the bottom of the body. So that's coming from the legs and everything below the heart, you know, the abdomen. And this is coming from the top of the body. So that's coming from your head and your arms and, you know, your shoulders, things like that. They both join together and they merge here. They both come in here. And this is the top chamber of the right-hand side. This is called the pulmonary vein on the left-hand side. And that comes from the lungs. Pulmonary means the lungs. So we've already learned the term like hepatic, which means the liver. We were talking about hepatocytes a moment ago. And we're going to start talking about pulmonary now, which means related to the lungs. So you've probably heard the term pulmonary disease before when people are talking about lung diseases. Pulmonary vein comes from the lung. So these are the veins that take the blood to the heart. And you remember it's veins in and artery out because there's an in the word vein. It says, if you look at it there, vein. So the vena cava is a vein. It's got lots of V's in it, so that helps us remember it's a vein. Vena cava and the pulmonary vein take the blood into the heart. So the pulmonary vein is taking the blood into the left-hand side, and the vena cava is taking the blood into the right-hand side. That's quite important because the blood is different in the right-hand side and the left-hand side. On one side, it's full of oxygen. The left-hand side is full of oxygen because the blood's just been through the lungs. And obviously, that's where we breathe in our oxygen. So the left side of the heart is full of oxygen. The right-hand side is empty of oxygen because that's just come back from what we call systemic circulation all the way around the body. So it's been around our entire system. And so it's got rid of all of its oxygen. It's given all of its oxygen to the tissues and to the cells. And I think we're pretty much out of time. So I will leave it there. I will come back on that slide next session. I think I'll go quickly over that again. Have you got any questions for me other than the one on uniporters and symporters, transporters? I'll get that slide back up again and we can go over it thoroughly. Let me just pop that chat up so I can see. If there's no questions, guys, have a good evening and I will see you on Saturday. Bye, everyone.